In the last chapter, we saw that the external vascular network of a young tunicate colony, which contains more than 30 ampulla and many sections of vessels connected in a complex net, contracts and expands every 60 to 90 seconds. These contractions are often irregular, but segments physically close to each other tend to be partially synchronized, implying interaction between segments, which could generate the complex contraction pattern. A simple system, for example, two ampulla connected by a short vessel, might beat in a regular way. Fortunately for us, more than 100 years ago, Frank Bancroft discovered detached ampulla continue to beat. If you assume an ampulla must have something to pump blood into, this is the most simple system. Each ampulla can exchange blood with the other if they contract 180 degrees out of phase. Follow the pair 2+3 near the top of the frame, made by cutting the connection below them to the rest of the colony. The field of view is a little messy. Remember, the ampulla are embedded in a thick tunic gel. Each ampulla is about 1/2 millimeter long, and I did this surgery by hand with an X-Acto knife. It's not so bad, but get somebody else to do your cataract surgery. This video is speeded up 20-fold so you can see the contractions. You can follow an occasional blood cell from 2 to 3 and back, but the cell concentration is low, and mostly you just see contractions. We need to look at the graph of area versus time to see what's really happening. That's what all my equipment and programming is for, after all. But let's just enjoy the movie for a few seconds. Holy sine wave. Those are two beautiful curves for two beautiful ampulla contracting 180 degrees out of phase. They look very much like sine waves, maybe not all the same height, but look, this is real biology, not maths. You know the drill from chapter 1. First, look at the Fourier power spectrum, then the phase portrait. As expected, almost all the energy in the Fourier power spectrum is in the main peak and small shoulders at a frequency of 16. Very different from the power spectrum of ampulla in colonies. As expected, the phase portrait is almost a collection of clean circles. I have plotted about 8 cycles here that are above the black bar in the original graph. You don't see any small loops or kinks, just nice big circles. Now for a change, we do something simple. Measure the times between peaks. If the contractions are regular, these times should be almost constant. Placed in the dish recorded a 3 degrees centigrade increase during the 24-minute observation time. Animals are very sensitive to temperature, and the observed increase in speed of contractions of 10% is about what you would expect for a 3 degrees increase in temperature. So the contractions are more regular than first appears. Up to now, I have implied, no, I've told you, ampulla are the units of contraction. All previous plots, calculations, and discussions were about areas of ampulla. If that were strictly true, it would not make sense to look for contractions of a single detached ampulla. How could the area of an isolated ampulla change? Where would the blood go if the ampulla contracted? But let's just look for the fun of it. None of these detached ampulla are connected by vessels to other ampulla. They are all singles. This video is speeded up 20x and you still see movement. What's going on? Let's zoom in to the third amp from the top. We see bulges that appear, move across the ampulla, and then dissolve. The blood cells inside are pushed by these contractions up and down the middle of the ampulla. This ampulla is not a single uniform contracting unit. Different parts contract with the same frequency, but different phases. Here is a plot of the total area of the ampulla. There are little peaks, but the pattern is messy, and the vertical y-axis shows the peaks have a range of only 1 or 2%. These small peaks only represent the degree to which the area of the ampulla doesn't measure its volume, since we know the total volume has to remain constant. I have scans of all 4 quarters. Let's look at the middle 2. They both have nice peaks throughout the 1,000 seconds, with a range of almost 10%, much greater than the whole ampulla average. The period is about 70 seconds, typical for ampulla attached to colonies. The second segment is out of phase with the third, most dramatic for the first 500 seconds. Now look at the first 2 segments. Segment 1 is very irregular and weak for the first 500 seconds, but then beats well, clearly not in phase with segment 2. I could bore you with plots of all the 10 remaining pairwise combinations, but I just summarize: no 2 segments contract in phase. Let's compare contractions of ampulla walls in an ampulla pair with contractions in the wall of a single ampulla. In the ampulla pair, the cells of each ampulla are in direct contact only with cells of their own ampulla. The connecting vessel isolates the 2 ampulla from most of the direct force of ampulla wall contractions because it is much smaller diameter, makes it relatively stiff. Cells in each ampulla only feel contractions of the other ampulla through blood pressure, not directly through cell-cell contacts. Thus, the cells in each ampulla can contract in synchrony, in phase, while the 2 ampulla contract 180 degrees out of phase, required to keep the total blood volume constant. In the single ampulla, all wall segments are connected by direct cell-cell contacts, and the phase necessary to keep the total volume constant has to smoothly change along the length of the ampulla. Thus, all segments are out of phase with all other segments to some degree. The unit of contraction must be the cell. Ampulla look like they were a unit only because all the cells contracted together, but in an isolated ampulla, the cells can't contract in phase together. They are forced to beat with different phases. The scales fall from our eyes. But where are the cells that I claim ampulla and vessel walls are made of? The only cells that we saw in the videos were blood cells, either moving through vessels or bound to walls. We don't see cells in ampulla and vessels because we can't see cell walls. Sheets of cells just look like messy gray space viewed face on, and since the cells are thin, look like thick lines edge on. I have made this crude drawing as a placeholder for the beautiful photo in the paper by Madhu et al. The cell walls have been labeled with green fluorescent antibodies to cadherin molecules, a protein which acts as bolts holding cells together. The cell widths are about 1/2 the diameter of the vessel, about 30 microns. Typical ampulla must contain 200 to 1,000 cells. This paper contains many other interesting results besides the image of the vessel, and the bibliography would be a good start for anybody interested in this tunicate system or cadherin. The process of two oscillating or rotating objects interacting to come to the same frequency and phase is called entrainment or synchronization. Entrainment usually implies a gradual incremental process, such as the pendulum of two clocks, slowly changing to the same phase due to the small vibrations in their common support. Synchronization is more general and could refer to oscillating electrical circuits that are always locked to the same frequency. The chatbot I consulted agreed that they are similar concepts, but claimed entrainment is more often used when the interacting objects are biological in nature. I don't agree, but who am I to differ? Maybe I was trained on different literature than the chatbot. However, we both agree that they are hot topics. There is entrainment of menstrual cycles in groups of women, planets with satellites, sleep cycles, firefly flashing, and on and on and on. There are many papers and books on the subject. One popular book is Sync: How Order Emerges from Chaos in the Universe, Nature, and Daily Life by Stephen Stogans. See a dramatic example here on YouTube. Search for 32 metronome synchronization. We have seen in Chapter 2 that each ampulla of an isolated pair beats in a regular, out-of-phase, sinusoidal wave to maintain a constant total volume. We have seen that the walls of a single isolated ampulla contract as a progressive sinusoidal wave to maintain a constant volume. I propose the cell to be the unit of contraction, with the phase determined by contact with neighboring contracting cells. We have seen how the study of a system that has been altered, ampulla surgically removed from the tunicate, helps us understand the original intact system. We now suspect movement is responsible for entraining cell contractions. Let's push things around and see what happens. Stay tuned for Chapter 3.